get it across. Yeah.
For the quizzes and stuff, we're writing scientific names, we need to underline them.
No, but you do need to write them, capitalize them properly. So I'm not going to ask you to underline the names, but I will ask you to capitalize the genus, capitalize the family, and the specific epithet, of course, is lowercase. Everybody's lowercase. So again, pay attention to that, and I'll remind you here and there. Does anybody have any questions about the about what I'm asking you to do? And if you do, and you just don't feel like asking, it'll become obvious later on. One of the, yes?
So it's just family on the 28th, and then scientific on the 5th of October?
Yes. Or both on October, on the midterm. I still want you to know the families. Don't forget the families. You still have to know that. And so one of the things I think that makes ichthyology a little bit more challenging than some of the other classes is, this is true for herpetology too, but ichthyologists and herpetologists communicate using scientific names, and that's just the bottom line. And part of that is because the common names for these animals are standard, but not standard across regions, and they're just not used consistently, especially when you're talking about non-scientists. So we communicate using scientific names. I wouldn't expect you guys to remember these for the rest of your life, but you probably will remember some of them, and if you come across them again, you're going to pick them up a lot easier. It's important that you know them, and I use them all the time. At the end of the year, I have to fill out a report on the animals that I collected, and part of that is writing the scientific name. And inexperienced people say, well, I can just look up the scientific name, which is true. But the problem with that is, anybody know what the problem with that would be? Maybe if I told you that, I go out in the field during the summer almost every day, every week, and we end up with massive amounts of names. And each one of those things has to be typed into the computer. I give the student technicians the data sheets to transcribe onto our spreadsheets, but I usually don't tell them to fill in the scientific name because that takes them forever to do that. Every name you go, look it up, every name you go. Where I can just look at the common name and write the scientific name. So I do that part just because I know the scientific names. It takes me a fraction of the time to do it. And so if you're doing that a lot, or if you're asking an agency person to do that, you're going to have to ask 10 people to do it instead of just one person. So it's important to know them. You're not going to know all of them, but you will learn a bunch of them, and you'll never forget them. If you're using them often, that's the way you communicate. And that's how fish biologists and herpetologists tend to communicate, is in the language of scientific names. So it's important to do it, and I think it's a good skill to learn. And again, the more you do it, the more you can apply that to non-fish or non-animals even, but a lot of scientific folks use the scientific names just because it's very consistent. Okay, that's my plug for learning the scientific names. So you just have to spend time doing it. It's not difficult, it's just something that you got to practice. And then I was talking about this, you also want to practice like you're going to play, right? So I'm going to ask you to write the scientific names on a sheet of paper. It's going to be a blank sheet of paper with lines. No, there's nothing on it. You got to fill it in with the family name and the scientific name. So when you're learning these things, practice like that. Expect to have to do it because that's what I'm going to ask you to do. And of course, if you do that and you find out you get it wrong, it's still time. Okay, so seriously, that's it. All right, so I'm just going to kind of relatively quickly go through the families, and I think for one thing that you'll see with this particular group is I'm presenting them to you roughly in the order of their evolutionary history. So these are the jawless fishes, so those are the earliest vertebrates, the jawless forms, and then we'll talk about more ancestral species, and then as we get closer and closer to the end of the semester, we'll talk about more derived groups. And so in some ways that kind of translates into the fact that these ancestral groups may not have many representatives anymore, just because everybody goes extinct eventually, right? So, so what you'll find in the first half of the semester is that you'll have a lot of family names and fewer species names within each family, just because there aren't many left. The second half of the semester, you're going to have fewer family names, but more genera and more species in each family, because those are the more recent ones and more derived, and there's still a lot of those still on Earth. And so it looks like a big list, and it's a list. I mean, it's not something to be taken lightly by any means. But if you understand this part of it, you really got the differences among the fishes, because almost all of them are in a different family. So, having said that, the first group I want to present is the family Petromyzontidae. And again, these names are not intuitive by any means, and so this is just going to take practice. So Petromyzontidae are the lampreys, and there's several different species, but their characteristics, of course, no movable jaws. They don't have paired fins, and I don't think you're going to mistake a lamprey for anything else. Maybe an eel, which you'll see today, but eels have movable jaws, of course, and they have at least paired pectoral fins. So the gill openings are multiple, and instead of having an operculum covering that common chamber, they've got several openings. They're elongated, their body shapes are different. There's different forms, so there's the parasitic form, which is this guy. They have these horny teeth that help them kind of grab onto a host, and then the tongue is what's described as a rasping organ, and it just rakes a wound on the side of the animal, the host, and that host will then either bleed or release lymph, fluid, and that's what they feed on primarily as adults, the ones that are parasitic. And this is a salmonid with some lampreys attached. And for the most part, most fishes are able to get rid of them eventually, and they're not a particular problem. In some cases, they become very abundant, or if they get into a place like they're not supposed to be, like the Great Lakes, then those animals that have evolved there did not evolve defense mechanisms for it, so they can cause real problems. But as far as their morphological descriptions, I think everybody's going to be able to recognize a lamprey when you see it. So you should be able to recognize the family Petromyzontidae pretty easy. In Tennessee, we have 5 species and 2 different genera. The Lampetra tends to be the non-parasitic ones, and then Ichthyomyzon are parasitic. And so in lab, I have 2. I've got the least brook lamprey, which is Lampetra apitera, and then the chestnut lamprey, which is Ichthyomyzon castaneus. And so they're fairly easily recognized one from the other. One is non-parasitic, and one is parasitic, so their mouths look quite a bit different if you look carefully at their mouths. There are other species, of course, and they're distinguished by the tooth structure. So the arrangement of the teeth and the oral disc is different among the species, and they also differ in myomere count. So we talked about myomeres in lecture. So those are the muscular bands that you see on the body wall. And let me go back. And so not real clear on this one, but you can kind of see it. The one that I have in the lab, you'll be able to see, but you'll see these bands of musculature all on the body wall, and you can count the numbers of those, and that will help you identify what species it is if you don't know where it came from, etc. Okay. Next group is the family Accipinceridae. This group includes the sturgeons, and together with the description I've given you, their worldwide distribution. So you can see that these guys are in North America, Asia as well. These guys have strongly heterocercal tails. Somebody want to tell me what that means? Generally speaking.
The vertebral column extends to the dorsal lobe.
Oh, yeah, perfect. So yeah, the heterocercal tail, the vertebral column kind of extends into the dorsal lobe of the caudal fin, so that's really obvious when you look at these guys. So this is the typical shape of a heterocercal tail. You can see the bony support, muscular support extends into only the dorsal lobe of the caudal fin. They have 5 rows of scales that are bony-like, scoop-like, and so on this guy, there's one here, and then one there, one on the other side, and there's a row down here on either side. So again, you're not going to confuse a sturgeon with anything else. They're morphologically pretty unique. They also have a fairly prominent snout, not as prominent as the next group, which are the paddlefish, but still a noticeable rostrum. They've got barbels on the underside of the snout. So this is an inferior mouth. So here's the mouth, under the underside of the animal, which is characteristic of these guys and some others. And this is what the barbels look like from the ventral surface. And so there's some variability among the species in the arrangement of these barbels. So sometimes they're in line, sometimes these two are set back. So that can help you distinguish one species from another. In Tennessee, we have 3 species and 2 genera, Acipensor and Scaphorhynchus. I've only got one specimen, Scaphorhynchus platyrhynchus, downstairs in the lab, so you only have one on your list from this particular family. These guys are distinguished from each other by their scalation, their scale patterns, and the barbel structure. And both of these animals kind of spawn in gravel beds and rivers and things, and so that by itself kind of makes them a little bit vulnerable to conservation problems, declining populations, because that kind of habitat is disappearing a little bit, or quite a bit in Tennessee and other parts of the country and the world for that matter. So they're experiencing some real serious threats to their populations because of longevity and age of maturity and their really specialized spawning habitat. Okay, next group is the family Polyodontidae. These are the paddlefish. And their distribution is disjunct, so we've got them in North America and also in Asia. They're closely related, of course, in the same family, but different species. They also have a heterocercal tail, and so you can kind of see it on this one, but not so good. Here's the dorsal lobe, and the vertebral column extends up into that dorsal lobe. They're scaleless, so they have smooth skin. They've got really tiny scales embedded in the skin in some places on the body, but mostly when you look at them, you don't see scales, so they're considered more or less scaleless. They have a conspicuous paddle-shaped snout or rostrum, and this is obviously that. And I think y'all saw that in the lab last time. So again, I don't think you'll mistake these guys for anything else. In Tennessee and in North America, we have one species, Polyodon spatula, and this is a photograph of one in the aquarium. They're planktonic. They feed on plankton. They're planktivores, and so this is an animal that's feeding. So they got really long gill rakers that are closely spaced, and they'll bring in large volumes of water and filter out the microorganisms, and then divert that into the digestive system. So, and the snout is covered with cells that are really sensitive to chemical cues, and so it probably helps them find patches of zooplankton in large volumes of water, like lakes and large rivers where they live. Okay, next group is the Lepisosteidae. These are the gars, and they're essentially North American and a little bit into Central America, but mostly North American group. They have an abbreviated heterocercal tail, which means the margin of the tail is symmetrical, but the internal support for the tail is asymmetrical, and so this is a pretty good picture of that. So the vertebral column kind of extends up into the top part of the caudal peduncle right here, but then it stops right about there, so that the outside margin of the tail is pretty symmetrical, but this part of it is asymmetrical, so there's more of it on the dorsal side as compared to the ventral side. So that's a characteristic of these guys. They have elongate snouts, so most of you guys have seen a gar, but this is pretty typical of what the gar morphology is like. The shape of that is a little bit different among the species, but they all have a fairly long snout, and they've got lots and lots of really sharp and rigid teeth. Mostly they feed on what, would you guess?
Fish.
Other fish, and that's typically an adaptation to feeding on fish, which are slippery and have scales that come off. They got long, pointy teeth that just get into the flesh, and animal can't get away once that happens. This is an alligator gar, so some of them get quite large, 10 feet or so. That's less common now than it used to be, but just because of conservation problems with the alligator gar and habitat destruction. But this is the largest species, and there's been records of them over 10 feet. And some of them are fairly recent records, but of course they're going to eat larger fish and whatever they can come across. Their bodies are covered with ganoid scales, so this is a pretty good indication. Although I don't think anybody will have any trouble with the family level distinction of these guys. And I was talking about this in lecture the other day. So their scales are rigid. They're kind of diamond shaped. They're not flexible at all. They're thick. and that mostly gives them protection from each other and predators. But it also makes them a little bit less maneuverable than the other fishes that have much more flexible scales. And it's lightweight, so they're expending a little bit more energy in moving around. Although, relative to most other things, they're pretty maneuverable. Okay, in Tennessee we have 4 species, in two genera. Atractosteus is the alligator gar, and that's this dude. And the main difference between these guys and the others is that, well, they get much bigger, their snouts are broad like an alligator, and the teeth, the distribution of teeth in the upper jaw are different in these guys than they are in the others. And so if you look closely enough, you'll see the differences in the mouth. And then the other 3 species are in the genus Lepisosteus. We've got spotted gar, long-nosed gar, and short-nosed gar. So here's a spotted gar. I don't think I have a short-nosed gar downstairs. I've got alligator and long-nosed. We do have short-nosed gar in Tennessee, and occasionally we'll collect them during the summer in some of the streams that we're working in. The most common one that we have in Tennessee, or at least in West Tennessee, is this guy, the spotted gar. So they got spots on the snouts and they've got spots on their fins, and you'll be able to see that when you see the ones that are preserved. And then the long-nosed gar, which is named because the snout is really long and skinny, slender compared to all the others, really. And that's the short-nosed gar. So the short-nosed gar and the alligator gar look similar, except these guys don't get nearly as big, and like I said before, the teeth, distribution of teeth in the mouth is much different. Okay, y'all can stop me if you have any questions. I feel like I'm really going through this fast, but we're going to go downstairs and look at the actual animals, so it should be okay. But if you have questions, just stop me. So Amiidae is the family bowfin. Includes the bowfin. Amiidae is the family. Bowfin is the species, and this is the distribution. So this is an exclusively North American species. There are no other species of bowfin anywhere on the planet, so that's kind of cool. They also have an abbreviate heterocersal tail. This is just a drawing of a typical bowfin. This is a male. The males have this distinct ocellus, which is dark in the middle and then usually orange on the outside of that. When they're breeding, their fins turn kind of emerald green, really pretty. And the males are fairly active in the parenting role, and they'll guard the babies as they get bigger and they move away from the nest, and they'll defend them and corral them and stuff. So it's kind of a neat fish. They have a lot of really interesting behavior. The characteristics of these guys include a really long dorsal fin. So for the species that we'll see, they have like the longest relative to their body dorsal fin. If you see something like that, it's definitely a bowfin. Yeah.
Are they like redfish where they get like different dots on the back? Or are they pretty much just one dot all the time?
You mean back here?
Yeah.
No, just one ocellus. Yeah, redfish have, like
They
big
get like
one, they
a
might have some
couple.
little one. Yeah. No, they're just that. I've never seen anything other than that. So I'm not 100% sure, but I think that's the case. But I know what you're talking about with redfish. They're different. Another thing about these guys that's unique, so you won't see any other fish in this lab that has it, and that's the gular plate, which is this thing right here. So it's right between the 2 halves of the lower jaw, and it's just a thick, bony plate. When I was in college, of course, I was doing the same thing you guys are doing, and the specimen that we had of the bowfin was about this big, and I didn't really know bowfin at the time, and so I said, okay, that's a bowfin. And then on the exam, we got a bowfin, and I looked at it and I'm like, what? I have no idea what that is. But I wasn't looking for the thing that distinguished it. I was just looking at the whole thing, and I didn't even bother to turn my to go look at that. Of course, it was a bowfin. I put largemouth bass, and I knew 100% that it wasn't a largemouth bass, but I had no idea what it was. So I just went with that and knew I was going to get it wrong. So kind of think about that when you're looking at these guys. It's like look for specific things, not just kind of overall shape, because that's going to mislead you sometimes. They also have tubular nostrils, and so the in current opening is on this tube, and then the X current is way back here. So what these guys do in the water, that sticks up, and they can pull in water, they sense the chemical components of the water, and then the water come out the excurrent opening. So that, like I was saying today, they don't use the water for respiration, they just only use it for chemical cues. But that gives them a pretty good sense of direction of where the chemicals are going to come from, kind of like a forked tongue for a snake. It allows them to do the same thing. So there aren't any other fish that you'll see, at least in West Tennessee, that have tubular nostrils like that. In Tennessee, worldwide, there's only one species, that's Amia cava. And you'll notice for a lot of these families and genera at least, Amiidae is the family, Amia is the genus. For the gar, Lepisostiidae is the family, Lepisostius is the genus. So some of the names are very similar, and if you can kind of remember that, then that's going to help you, remember it on the final exam or on the exam. Okay, next group is the family Hyodontidae. These are the moon eyes and golden eye, and I don't know if I've ever seen a living one of these. I know they're relatively common. I just don't do a lot of collecting on lakes and big rivers, so I haven't really seen very many. But overall, they are relatively large fish. They're silvery or kind of gold silvery. They all, both of the ones that we have a narrow breast and belly. So we talked about the basic morphology of fishes. So the breast is just behind the gills, and the belly, of course, is posterior to that. For these guys, it's narrow. They also have adipose eyelids, which are, so adipose is fat, and so they've got these eyelid or structure that kind of covers the eyeball. It's kind of hard to see in the preserved specimen, so I don't think you really need to rely on that, but it is a characteristic of this particular family. They do have an axillary process, and it's kind of, oh, that's not what I said. On the pectoral fin, so here's the pectoral fin, and then so here's the fin, and then this axillary process, it's like a separate component to the fin. That's pretty obvious to see. So there are a couple of other groups that have an axillary process on one of the fins, but in combination with the other characteristics, I don't think you'll misidentify these guys if you're paying attention to that. If it has an axillary process and some of the other things that are obvious, then it's definitely the family Hyodontidae. They have cycloid scales. You guys didn't get a chance to see those in lab, but I've got them upstairs and I'll bring them down so you can look at them this time, just so you can see what that looks like. This family is in a bigger group that we'll talk about in class called Osteoglossomorpha. So osteo means what? Anybody know? Osteo? Yeah, osteoporosis. Tara said it. Bone. Right? So osteo is bone. Glossa refers to the tongue. Morpha is morphology or form. So these guys commonly are called bony tongues. Another example of the bony tongue is Arapaima. Anybody ever heard of an Arapaima, Arowana? Those are bony tongues. And they have teeth on their tongue. So here's the tongue, and you can see the teeth. And in the lab, of course, if you open up the jaw and look at the tongue, it's got teeth on it. So that's kind of unique. And they usually, they'll use the teeth and mash their prey up against the roof of the mouth, and that's how they, ultimately dispatch of prey that they eat instead of jaw teeth. They don't have jaw teeth; they have teeth on their tongues. Okay, so in Tennessee we have two species: Hyodon allosoides, which is this one, and then Hyodon turgisus, which is this one. So they look pretty similar. So you can see if you looked at it, it's like, yep, that's the same fish. All fish look exactly alike. But if you look carefully, they've got some features that are different, and it's a little bit. I'm not sure what was. Yeah. So they're distinguished one from each other by this keel. And so I said they have a narrow breast and belly. It's narrow in the sense that it forms a keel-like structure. So a keel, like a keel on a boat, where it comes down to a sharp V. And the nature of that is different if you look at one of them compared to the other one. So this is Hyodon turgisus, and you can see that the keel goes from the pelvic fin, really from the insertion of the pelvic fin to the distal end of the pelvic fin, and so it's relatively short in other words. In Allosoides, this extends way up into the belly and much further anterior. And so I like to say Allosoides goes all the way in reference to the keel. So the keel in Allosoides is much longer. So overall, they look exactly the same, but if you look at something specific, then they're very different.
So the picture is Turgus's?
Yeah, this is a Turgus's keel. This is the belly of the Turgus's. So it's a relatively short keel, and in Allosoides it extends further anteriorly. It's pretty obvious when you look at them, but they do look alike. I mean, they've got very similar morphologies otherwise as well. And there's some differences in their coloration too. In life, they're a little bit different in color, but once you put them in, preservative, they look the same. Okay, next group, family Anguillidae. These are freshwater eels. I don't think anybody's going to mistake this for anything, but they're elongated, similar to a lamprey, except lampreys don't have paired fins. What these guys do, they have pectoral fins. They don't have pelvic fins, so that's a little bit unusual relative to the other fishes. Of course, they have jaws, movable jaws. I'm basically distinguishing these guys from the lampreys. They have a long dorsal fin that's continuous with the caudal fin and continuous with the anal fin. So basically they have one big medial fin. It starts on the dorsal surface or ends on the dorsal surface. So it goes from the dorsal all the way to the belly. Just one medial fin as opposed to having a dorsal fin, a caudal fin, and an anal fin. It's all joined together. In Tennessee, we have one species, Anguilla rostrata, American eel. And so again, you can see the family is Anguillidae, the genus is Anguilla. So let that help you learn the names of the species generally. Okay, next group is the family Clupeidae. This includes the shad and the shads and the herrings. There's a lot of species. Some of them are marine species, a lot of them are marine, but there's a few that are mostly freshwater, and that's kind of what we're focused on. Generally, like a big one of these looks a little bit like the hyodon, so sometimes people get those confused. But I'll show you some things that are different among them. One of the things that's not different among them is that these also have a keel, a keeled belly. So, but the scales form kind of a sharp tooth or serrated keel on the belly. I don't think I have a picture, but when we get down to the lab, I'll point it out to you. You can actually feel it. If you run your finger from the back to the front, you'll definitely feel the serrations, which is basically where the scales come together and there's a little space between the scales, and so that makes a serration. But other than that, it's kind of a sharp keel on the belly, just like the hyodon. In Tennessee, we have three species. Dorosoma sapedianum, that's the gizzard shad. That's the most common fish I see in Reelfoot Lake, or even in the streams that we sample, we'll get gizzard shad sometimes. But occasionally we also get the threadfin shad, which is Dorosoma petinense, and then one that I never see is Alosa crassicloris. This is the skipjack herring. I think that's more common in like rivers, bigger rivers and lakes. You guys ever see the skipjack?
Yeah.
If you find one and put it in the cooler, bring it out. I could use a few more specimens. Okay. Yeah, I'll take it. It's in good shape. When we go electrofishing at Reelfoot, we'll get hundreds of these guys. Most of them are small, but and they get pretty big, 20 centimeters or so, 25 centimeters. The main difference among them, well, this one, what's the main difference of this one compared to these two? What's that? Yeah, they're not as deep bodied, and so longer relative to their height. These two are shorter and deeper bodied. In reality, this, as an adult, this one gets really big, this one stays pretty small. But that's hard to tell if you've got a small gizzard shad and a small threadfin shad. The main difference is the spot on the threadfin shad. The spot is relatively small compared to the gizzard shad. You can't really see it on this animal, on this picture, but when we get down to the lab, you'll be able to see that. The other thing that's different about them is this guy has a mouth that's terminal, it's right on the tip of the snout, and this guy has a mouth that's just a little bit below the snout, which is called what? Subterminal. So, and again, if you just have one of them, it's a little bit hard to tell, but if you have both, it's really easy. I see now. But that's probably the quickest way. They both have this really long last few elements of the dorsal fin. So of course this guy's called thread fin because of that, but this guy also has that feature, so that won't distinguish one from the other. But the best way to do it is to look at the spot. This is called a humeral spot and the position of the mouth. Of course, you know, I'm putting these fish in jars, and so I usually try to find small ones to fit in the jar. So the gizzard shad that we have is going to be relatively small, but I think you'll be able to distinguish them. Okay, next group is the family Catostomidae, and these are suckers. Is this on the slideshow that I gave you?
Yes.
Okay. Not sure where I stopped. Okay, so suckers, of course, suck. And the reason why they do that is because they're feeding on benthic invertebrates for the most part. And so one of their main features is that they've got a more inferior or ventrally facing mouth. It's usually fleshy, so the lips are fleshy. Mostly it's because the lips have what are called papillae on them, so a lot of grooves and folds, and that has a lot of chemosensory cells in it, so it helps them as they're moving around on the bottom find mussels instead of muck. Right? So they're not eating the mud, they're eating invertebrates that they come across, and they can kind of taste them with those papillae on the lips. Let's see. So I don't think you'll really have a problem with these guys, but some of the smaller species of suckers might resemble the minnows that we'll talk about in a minute. Or I think they're on this list. But some of the things that are different about them is that the anal fin in suckers is usually further back than in the shiners, so that's one of the things that's different. But mostly the suckers are much larger. There's also some differences in the dorsal fin ray counts and things like that, so they do have some morphometric differences among them, but one of the main things is their mouth position and their size. In Tennessee, we have 21 species of suckers, so fairly diverse in Tennessee. They tend to be more medium to larger river species, and so of course we have a lot of that habitat, not so much in West Tennessee, but in other parts of the state. So pretty good diversity of suckers. Some examples of the ones that we do have. This guy's a spotted sucker, that's on your list. The main thing of the spotted suckers is they have a little spot on each one of the scales. The one in the lab is a little bit harder to see, so you kind of have to turn them a little bit to see that, but it's there, so just kind of pay attention to that. This one is a northern hog sucker. We do have them in West Tennessee, but you have to go a little bit further south to find streams that have a little bit more gravel in them, because that's their preferred habitat. Faster moving streams with gravel bottoms, so we just don't have a lot of that in northwest Tennessee. This is one of my favorites. This is one that we collect quite a bit in several streams in West Tennessee. This is a black-tailed red horse, one of the suckers, and you can see that this one's probably a male spawning. So the main characteristic is this real distinct black mark on the caudal fin, and then it's the lower rays of the caudal fin are without pigment. So really contrasting black and white, and then we've got some red on there, and then when they're spawning, the fins turn bright red. So they're really pretty animals. They get, over a foot in length. That's their maximum size. Some of the suckers get pretty large. Okay, so one thing I should mention, so the family Leucicidae includes the shiners and minnows, but that family, up until relatively recently, was the family Cyprinidae, cyprinids, and so I think I've kind of referred to cyprinids in an earlier slide. So that family has been split apart into two families, cyprinids, which are now mostly European group, and then the North American members of that group are now in a different family, the family Leucicidae. So in the lab, you're going to see a jar that says family Cyprinidae, species Luxulus chrysopephalus or something like that. That family is no longer Cyprinidae. That's going to be Leucicidae. But I don't want to go through and change all the names. figured it'd just be easier to tell you guys. So anyway, so leucistids equals cyprinids for the most part. Okay, that says shiners and minnows. Feels like it would say something else, but it's probably what shiners and minnows. Okay, so these are mostly small species, fusiform body shape. That just means bullet shape, so the standard fish shape basically is what that means, as opposed to being elongated or rounded or something. So they're typically just kind of basic fish-shaped animals. They have either like deeply emarginate or forked tails, pretty characteristic of them. And so this is the basic shiner morphology. There are some that deviate from that a little bit, but that's the basic shape of a leuciscid. One of the things that's true about them is they don't have teeth in the mouth. It's not unique. There are some other species of fish, like the bony tongues, that don't have jaw teeth, but they do have pharyngeal teeth. And this is a pair of pharyngeal teeth that have been removed from an individual. And those are kind of on the back of the throat near the pharyngeal openings. And they use that to crush their prey. So they'll pull in a mayfly and then they'll use those teeth to crush this way, and then they'll divert that into the digestive system. So that's what they use it for. The other thing that's important about pharyngeal teeth is that it's very useful in determining species. They're species specific in combination with some other things. And so in some of the keys, for keying out leucistids, the first thing you do is remove the pharyngeal teeth and determine what their arrangement is. In fishes of Tennessee, that's not the case, but it's in there depending on what path you take. But we haven't really had the need to do that for the animals that we come across typically. But in Louisiana, I spent a lot of time keying out cyprinids and a bunch of other fishes that were collected throughout the state, and that was the first thing I did for every single one of them. So I got pretty good at it, but and of course then you don't have to do it every time once you figured out where it is. But that was like the first couplet, what the pharyngeal teeth arrangement is. And so you can see on this one, there's usually 4 on the outside edge of that pharyngeal bone, and sometimes there's none on the inside edge. Sometimes there's 2. Sometimes there's 1. So whatever combination that is, and that tells you to do this, and if it's a different combination in the keys, you go somewhere else. But for the fish, they're using it to crush their prey. For ichthyologists, we use it to identify species. Okay, so there's a bunch of species of these. So obviously, I'm just a small fraction of the ones I'm going to ask you to look at. These are some that we've come across pretty commonly. This is a bullhead minnow. This is one of the pineapple leaves is the genus. This is one of my favorites. This is a tail-like shark. And I don't come across these very often, but I've collected them out of Real Foot Lake, but just real sporadically. I've collected them down in probably the Hatchie. I think that's where we were. It's been so long ago, I'm not sure where we were. Andy Slider took me down there, and I have no idea where we were at that time. I just got into Tennessee, and he said, "Come on, let's go down." So I don't know where we were, but I think that's where we were. And so they're kind of open water species, but this is a male, and you can see how pretty they are when they're spawning. But they have this real distinct spot right here on the back of the caudal fin. And I've got one in the lab that you guys are going to look at and learn.
What was the name of that last one?
That's a tail-like shiner. That's Notrochus maculatus is the sign. Okay, next group is the family Umbridae. These are the mud minnows. They're small, elongate fish, look just like a minnow. Some of the things that are typical of them is they have a really short snout. So on a fish, the snout is between the eye and the tip of the end of the body. So you can see that their snouts are really, really small. They have a dorsal fin that's posteriorly placed, so far back on the body. That's pretty typical of these guys. Another thing that's a little bit unusual about them is they don't have a lateral line system. So when you look at like a bluegill, you'll see this kind of row of scales that's got holes in it. Distinct, if you notice that lateral line. These guys don't have a lateral line. And there are some other species that don't, so it's not completely unique, but it's true for this group. And so you'll see that some of the darters don't have a lateral line, or only have a few pores, but most of the group have distinct lateral lines, so that's a little bit unusual. In Tennessee, we only have one species, that's the what is it? Central mud minnow. Umbra limi, again, the family is Umbridae, the genus is Umbra, so that'll be helpful. These guys, of course, this is not what they look like in West Tennessee because we don't have water like that, but they're real common at Reelfoot Lake. So when I do the Tennessee Naturalist program, I go to one particular place because I know I'm going to get a bunch of different swamp species, and this is one that is always there in that spot. We just pull the same through all the vegetation, and we get mud minnows. We also get them in streams too when we're doing our stream surveys, so they're pretty common. They don't school, so they tend to be like one or two. We don't get a bunch of them, but they're consistently in the same place. They're pretty neat. The next group is the Aphrodidaridae, the pirate perch. This is again relatively small fish. They have a pretty large mouth for such a small fish. It looks kind of like whoa, gosh, what I don't know how to make it go. Let's see if I push one of those. I did it. I learned something today. Sweet. Okay, so yeah, it looks like they've got kind of a small mouth, but the back of the jaw kind of extends down here. So for a small fish, they have a relatively large mouth. They're deep bodied, but kind of diamond shaped. So the deepest part is right in the middle. So they're like like that, which is kind of not unique again, but diagnostic, I guess. When I'm out in the field looking at stuff, that's one of the first things I notice if I pick up a fish that's flopping around. I can look at their shape and kind of not identify them necessarily, but indicate one thing or another. They have spines on the dorsal and anal fin, so most of these are raised, but they do have one spine on the dorsal fin and one spine on the anal fin, so that's a little bit different. Most of the ones that have spines have more. These have fewer. One thing that's really unique about these guys is the anal opening is in what's called the jugular position, which is the throat. So on this guy, the anal opening is going to be right here behind his head. When they're developing, the anal opening is in a normal position, which is back here. But as they develop, that gets reorganized, and so that the digestive system does this. It goes from the esophagus into the stomach, and the intestines come up here, and it empties from the anal opening that's jugular in position. And I guess the best explanation that I've read is related to their nesting ecology, because they live in kind of slow-moving, swampy environments, still water with relatively little oxygen, and they nest in like crevices and spaces on the banks of those places. And so, of course, if you're backed up into a hole and you have your eggs there, and your anal opening is right over the eggs, every time you release waste products, it goes directly onto the eggs. And so what they do is that their digestive system opens up at the front end, so that stuff gets excreted and gets washed away downstream instead of piling up on the eggs, which will, of course, limit the amount of dissolved oxygen that the eggs are going to get and kill the eggs. So in Tennessee, we have one species. It's the pirate perch. I like to call them irate pirate perches, just because that's hilarious. And they get pretty irate when you electrocute them. It's no exaggeration. They're mad. And I can tell by looking at them. The genus is Aphrodidarius, and the specific epithet is Cyanus. And so traditionally, every time someone says Aphrodidarius cyanus, everybody in the class says anus. So I expect that from you guys, because you guys are all about tradition, I hope. Okay, I was going to say one. I think I was just going to refer to the. Are y'all getting tired of me saying this? Aphrodidaridae and then Aphrodidarius. Same. Are y'all getting tired of me saying that? I'm just trying to convince myself that ichthyology is not hard, but it is. All right, so family Elastomathidae are the pygmy sunfishes. These guys are awesome. I love the pygmy sunfishes. Again, this is southeastern U.S., so exclusively North American fish. They're very small. They have a single dorsal fin with spines and rays, just like the sunfish. This group has been combined with the sunfish over their taxonomic history, but more recent evidence suggests that they're a completely separate group. Still, you know, more or less related to the sunfish, but not in the same family. And there's differences in the scalation and their spines and rays and scale counts and lateral lines and stuff. So some of the things about them, they've got a short rounded pectoral fin, so you can see like a bluegill or a redbreast has got this really long kind of pointed pectoral fin. These guys have mostly rounded pectoral fins and it's short relative to the width of it. They don't have a lateral line, so that's different. They're totally awesome, of course. This is a male in breeding condition. This one doesn't even look that fantastic, but I used to teach ichthyology in the spring, and we would go up to Ovine River where it crosses the highway, or it goes under the highway. Sometimes it crosses the highway, but we don't go up when it's doing that. But in those ditches right before you get to it is where these guys were. In the spring, they breed in March, and so you go out there and collect them, and they'd be all fired up and stuff. And that's the males that get really elongate, feathery dorsal fins and anal fins. And then they also have a prominent what's called humeral spot, so that's another thing that kind of distinguishes them from some of the others in the shoulder area. It's fairly noticeable. Okay, geez, what was the last group?
We're on 18 and
Good
21
golly.
slides.
Okay, so family Piscileidae, I don't know, is this helping or not? I don't know. Okay, so the family Piscileidae, these are commonly referred to as the live bearers. It includes fish like black mollies, I've heard of mollies, guppies, that animals you get in the pet shop. Those are all in the family Piscileidae. We have a member in Tennessee, that's this guy, the mosquito fish, Gambusia affinis. They're small, of course, with superior mouths, and you can kind of get the sense on this guy, so the mouth is turned upward. They feed on a variety of things, but they feed on the surface. They also feed on mosquito larvae, which is why they call them mosquito fish. They've got a teardrop under the eye, which is not unique, but it's characteristic of the family of this particular species. This is different relative to the others. The males have an intromittent organ that they use to transfer sperm. So they're live bearers, which means that the sperm has to get into the reproductive tract of the female, and the gonopodium is used to do that. And this is a slide showing the gonopodium. And so the anal opening is here, and so so essentially what that is the anterior most anal fin rays are elongated and form like a little tube. And then when the animals are mating, this gonopodium swings forward, and that is inserted into the genital opening of the female, and sperm is released and fertilizes the ova. This is what the female looks like when she's gravid or got developing embryos inside. So you can see this spot here that shows up. That's basically the peritoneum, or the lining of the body cavity. As the abdomen expands, that becomes visible. It's kind of a signal to males. Basically, like I'm already accounted for, so I don't even bother. Not that probably discourages any of them, of course. But that's just the way they are. I think that's all I can say. Okay, another group that's similar to these guys are the top minnows, Fundulidae. They're also North American. They also have a superior mouth. What they don't have are gonopodia. Right? So these guys are not live bearers. They lay eggs and have external fertilization, so they don't have that. But they have many of the other features that I described. They have prominent teeth on the jaw. So this is a group that's in a bigger group called Cyprinodontiformes, which are the toothed minnows. And so that's what that refers to, but in the family Fundulidae. They have dorsal fin that's posterior, just like the mud minnow, so they're a little bit similar in shape to those guys. They have a rounded or truncated caudal fin. In Tennessee, we have quite a few species. The ones that we find around here are much fewer than that, but they're all in the genus Fundulus. This is one that, well, this one's one we have here. This is a golden top minnow. They're pretty common at Reelfoot Lake. Occasionally I'll collect them in streams. But the place I've seen them most abundantly is at Reelfoot Lake and thickly vegetated places. And they're really pretty when they're spawning too. They've got these really bright red spots and golden specks, and so they're kind of cool to have in the aquarium. This guy is, we don't have him in West Tennessee, but we have him in Middle Tennessee. This is a northern studfish. And this, of course, is a breeding male, so they don't really look like that typically, but they get pretty big. I've caught these guys on hook and line before, so they get, maybe 5 or 6 inches long, and they're pretty aggressive at feeding. So you might catch one day if you're fishing a little stream like Hurricane Creek or Standing Rock Creek. That's where I've caught them on hook and line. Then this is the one, or let's see this now. This is the one that we get mostly around here. This is the black spotted top minnow. And this is one of its sister species. This is the black striped top minnow. I think I just have this one downstairs. So they look pretty similar, and if you're not really careful, you can kind of misidentify them. But they're also pretty common at Reelfoot Lake. And so on our trip, when we go overnight, we'll stop at some of the boat ramps and seine at nighttime, and we'll collect some of these guys. This is a starhead top minnow, also at Reelfoot Lake. So you kind of get the sense these animals they're mostly still water kind of fishes. These two particularly live in streams, but not in the main current of the stream. They like the backwater, places that have a lot of vegetation. They have superior mouths, so they're feeding on lots of stuff falling in from vegetation around the stream. So you're not really going to find them out in open water because there's nothing really falling from onto the surface in the open water. Is this on the list?
This one, yeah.
Yeah. Okay, so Athyrinidae, these are the silver sides, and so some of their characteristics, they're flat topped, which is typical of them. They've got pectoral fins that are high up on the sides, and we'll talk about that in the lecture a little bit. There's a kind of an evolutionary change in position in some of the fins, and that kind of represents that. So pectoral fins are high on the side. They've got dorsal fins that are widely spaced. So that's pretty unique compared to all the other ones that we'll look at. Some of the fish that you'll see in lab later have a notch between the spiny dorsal and the soft dorsal, but these guys have a space. So widely spaced components to the dorsal fin is a pretty good clue that it's Athyrinidae. They've got cycloid scales too. In Tennessee, we have 2 species, Labidestes siculus, which is the brook silverside, which is this guy, and then Menidia berylina, which is the inland silverside. Both of these I've collected at Reelfoot Lake. This one tends to be more of a river species, but we might collect both species at the lake when we go out there. This one is the most common one that we get around here, at least the ones I've come across most typically. You guys might see the inland silverside quite a bit. Okay, last family. Family Associates. This is the pikes and the pickerels. So their bodies, their body shape is kind of elongate. They have a kind of a flattened snout, like a duck bill kind of shape. Their body is circular in cross section. So if you cut them in half and you look at it from the end, they're kind of a round cross section. They've got elasmoid scales, which are the flexible ones, as opposed to the gars, which have the ganoid scale. So you might look at that and say it looks similar to a gar, but the scales are much different, the snout's different, the fins are different. So if you're looking at the right thing, they're pretty easily distinguished. So we have 3 species in Tennessee: Esox americanus, which is a redfin pickerel; Esox niger, which is a chain pickerel. Oops. And then that's the other one is Esox, what is it, guys?
Say it again.
The other Esox that we have in Tennessee.
Oh, muscle.
Yeah, Esox musquinongi. The muskie. I don't know why I don't have a picture of it, but we do have that in Tennessee. Okay, that's the end of the slideshow. Does anybody have any questions? Yes, sir.
The pickerels relative to size of the gar.
Small one, typically. So the redfin pickerels don't get very big. The chain pickerels get bigger. It's the same family as pike and muskie, so they get big. But we don't have.
Yeah.
Okay. So